Okay, very good morning, students.
Good
Welcome.
morning, ma'am.
Yes, welcome to biology classes. Today we are going to see the next part of chapter number 6, that is molecular basis of inheritance, right? In the previous video, we have already completed and covered so many points, right? Today we are going to start with the next topic, and that is the HGP, that is Human Genome Project. Okay, now in the previous video, we have seen and covered about the different genome sequencing, right? So now today we are going to see the goals of HGP, right? That how the goals are taking place, right? Or what are the goals of Human Genome Project. So first is the particular, okay, first is the first goal. See, yesterday I told you about the bioinformatics, right? The bioinformatics means it is for the storage and receiving and analyzing this data. A new branch of biology has been developed, right? So bioinformatics is a branch of biology in which what happens, there is a storing of the information as well as the receiving of the information, and how to analyze the data in a given particular sequence, right? So this all things are occurring in a branch which is known as bioinformatics. Now, this bioinformatics is very useful in understanding the different types of sequences of the animals, not only of the humans, but also of the different types of animals, right? So now this particular bioinformatics is also leading to the Human Genome Project, and this Human Genome Project, this project is also including several animals also, right? That means if you are looking at the human's genome, right, then means all the genes which are present. So the genes, all the genes combine to form the whole sequence, right? So the whole genome of the human, when it is learned or when it is find out, what they found is they found that 3,300 books were written, and this was comprised of the whole human genome. And this all the 3,300 books, they all were what, the of 1,000 pages, right? So now we are going to see that what are the goals of HGP. So the first goal is, so the first goal is to identify all the approximately 20,000 to 25,000 genes in human DNA, right? So in human DNA, what you can see is you can identify, right, the all the different types of genes. That is, there are total 20,000 to 25,000 genes which are present in the human, and this all the genes are identified, right? So that is all thing which is possible due to the HGP, that is Human Genome Project. The next is, it determines the sequences of the 3 billion chemical base pair that make up human DNA. Now, to make up the human DNA, what you need to do is you need to do the sequencing of the 3 billion chemical base pairs. The base pairings are what? The adenine binds with the thymine, the guanine binds with the cytosine, and the final result of all these things is the protein which is formed at the last, at when the process of translation completes, right? So to determine the sequences, that is the 3 million or 3 billion base pairs, chemically this Human Genome Project is used. Next is, store this and stores this information in databases. Right now, this through the use of bioinformatics, you can do what, the storing of the particular all the databases, right? That means the binding or the corresponding attachment, that is the base pairing of the nucleotides which is occurring, right? That is, the adenine binds with the thymine, the guanine binds with the cytosine, right? Just a second. Okay, so next is the next goal of the HGP is to improve tools for data analysis, right? So it is also going to do what? It is going to improve the different types of tools which are used, and they are for the data analysis. That means how to analyze the data in a proper manner, and what kind of data should be involved in this type of Human Genome Project. So with the help of the Human Genome Project also, you can improve the different types of tools which are used to understand the human genome. Next is the transfer related technologies to other sectors like industries, right? So in industries also they are using several bacteria, several yeast, right, several fungi. So they need to understand and they need to know whether what kind of genome sequence they do have. So for understanding the genome sequencing, what you can do is you can, the industries are using this kind of human genome projects, like the genome sequencing, right? So they are using this type of technologies to understand the whole genome sequence of a particular fungi, bacteria or anything, right? And upon understanding this genome, they can easily find out the exact mechanisms which are taking place through those bacteria, fungi and else animals or the microorganisms, right? Next is, and the last point of the goals of HGP is that they address the ethical, legal, social issues, that is ALSI, that may arise from the project. Now, through this project, there are so many things which are taking place, that is ELSI. ELSI means the ethical, just a second. Okay, so they are used in ethical ways as well as in the legal ways also, and the social issues are also being demonstrated, or they are also being where they are being used by this human genome project. So all these things are kept in mind while doing the human genome project, right? So this is all about the uh, you can say the goals of HGP. Now, the next is the methodologies which are used during the human genome project. So there are two methods of human genome project, and or we can say there are two methods which are applicable, right? The first method is the ESTs, right? The full form of ESTs is expressed sequence tags, right? And the second one is the sequence annotation, right? The first is the ESTs. So we are going to see the first one, that is ESTs, and it refers to the part of the DNA that is expressed. So whatever portion which is expressed, that is said to be the particular expressed sequence tags, right? Next is, and here it is transcribed as mRNA and translated into proteins, that is the different types of proteins, we can say, right? So here ESTs are the sequences which are said to be the express sequence tags, right? That means those sequences which are only expressing, right? So when the expression is taking place, what happens, the DNA gets translated, the DNA gets replicated and it forms another DNA, that is the newly synthesized DNA. From the DNA, the mRNA is formed, that is the transcription, and after that the translation takes place and it forms the protein from that particular mRNA. So that is nothing but the sequencing which is taking place, and it only reads the frame, only reads the express sequences, and for this reason the first method is to read all the expressed sequencing tags, right? So the first method is ESTs. The second method is sequence annotation, right? Sequence annotation says that in this method the entire genome, that is the coding as well as the non-coding strands, is coding and later on function as assigned to the each region in the genome. So here the difference between the sequence annotation and the second, that is the ESTs, the difference between them is in the ESTs only the express sequences are taken, but while we are talking about the sequence annotation, they they take both the strands, or we can say both the regions of the DNA, that is the coding strand as well as the non-coding strand, or we can say the coding regions as well as the non-coding regions, right? So they are taking, in this means in the sequence annotation, these two of the regions, that is the coding as well as the non-coding regions, the two of them take part, and these two of them when they are combined, and at last what they do is at last they are combined to perform a new function, right? So these are the two methods which are useful in the human genome project or while carrying out the human genome project. Now from this human genome project we can understand that there is occurring the process which is occurring, that is the genome sequencing, and the genome sequencing which is taking place is is what it is in it. So that is the DNA from the cells is isolated and is randomly broken into fragments of smaller sizes. So here what happens, the particular DNA from the cells, so from the cells the DNA is isolated, so the extraction of DNA occurs from the cells. And when the cells are the the DNA which is present inside the cell, when it is extracted out or isolated, what they are doing is they are randomly breaking that DNA into smaller fragments of smaller sizes. So the first thing which is occurring in the genome sequencing is they are isolating the DNA from the cells, and after isolating, what happens, they are going for the breakdown of the particular different types of the DNA into smaller fragments. Okay, so next is the fragments are cloned into the suitable host using vector. Now using the vectors, right? Vectors are the vehicles which are used to transport the DNA from one organism to the next organism, right? So for transporting, okay, so the fragments are cloned into the suitable host, right? So now the cloning process means the duplication which is taking place. So the copies which are formed, it will be forming the exact copies of the parent cell, right? So from the parent DNA, it is going to form the uh, exact things, and when it is forming the exact copy of that parent, what they are doing is those fragments are using the vector, they are transferring into the host, right? Now the clone segments amplify in the host, right? Now the clone segments, they get amplified, right? Amplified means to do the amplification, the doubling of the DNA which is taking place, right? So first what they did is they isolated the cell, they took the cell, they isolated the DNA, and upon the isolation of DNA, what they did is they broken down the DNA into irregular segments, but they were smaller in size. So now we are having the different types of fragments of DNA, and when the segments of DNA are formed, what happens, they are being inserted into the vectors, and these vectors are transferred into the host cell. Now the DNA is already transferred into the host cell, and now in the host cell, this cloned sequences, that means the sequences which are the gene of interest, because all the sequences are in the host organism, and in the host organism they are going for the duplication of their own self, that is the amplification is taking place, and that type of amplification is known as, it is occurring through PCR, that is polymerase chain reaction, which you are going to study in detail in chapter number 12, right? So here in this particular, what happens, the clone segments, they amplify in the host, and the amplification facilitates an easy sequencing. With the use of this amplification, what it is occurring, the amplification, it helps to produce more copies of itself, just like the previous one or just like the parent one, and it becomes very easier to sequence the whole genome, right? Next is the common vectors used, which are used are, the first one is the BAC, that is bacteria artificial chromosome, and the second is YAC, that is yeast artificial chromosome. So there are two different types of common factors which are normally used, right? And that is, first is BAC, that is bacteria is used, for this reason it is known as BAC, and if yeast are used, then it is known as YAC. So it can be asked in the SQ questions that which are the vectors or which are the common vectors which are used in the genome sequencing. So that is the two, that is bacteria artificial chromosome, and the second one is the yeast artificial chromosome. Now the common host are bacteria and yeast, as the two of the common vectors are of the yeast and bacteria. For this reason the common host will be also the yeast and bacteria. So the particular host will be bacteria as well as the yeast. Next is the automated sequences are used to sequence this smaller fragments by Sanger sequencing, right? There is a process which is known as Sanger sequencing, through which all the automated sequences are sequenced into smaller fragments. So they are arranged into small fragments, and it occurs and it forms a sequence like structure, and that is occurred by the process of Sanger sequencing, right? Next is the sequence is so obtained are arranged based on overlapping regions within them. Now here in some regions there may be chances that there can be a overlapping which is taking place, right? So you should be very careful about it, that while doing the sequencing there should be no overlapping of the sequences with each other. So the sequences which are obtained by carrying out the particular genome sequencing, what they are doing is it is always based on the overlapping regions, right? That means there will be some overlapping, that is if one strand is present and upon that strand another strand is present, so there will be a overlapping which is taking place, right? Next is the alignment of the sequences is also done automatically by computer programs, right? Now here, as the bioinformatics is also linked with this particular types of information, what it is, the alignment or the arrangement of the particular sequences is also done automatically. That means they are not going to check whether which type of sequences are entered or whether it is present on the wrong side or not, right? So here, first of all, they are going to automatically, the computer programs are going to recheck it, and they are going to use it as for the genome sequencing. And the last point of the genome sequencing is, then these sequences are annotated and assigned to each chromosome, right? So after this, the sequences, they are annotated, that is the coding as well as the non-coding regions, they all are going to be assigned with each other, and they are going to be present on each chromosome. That is, each and every chromosome will be going to contain two types of regions. The first will be the coding regions, and the second one will be the non-coding regions. And based upon all these things, they are going to do what? The sequencing of the genome, right? So this is all about the genome sequencing. And the next portion is the genetic and physical maps on genome. Now we know about what is genome. That is, so many genes are combining with each other to form a sequence which is known as a genome, right? So this genome, now this was generated using information on polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellites, right? So the genetic and physical maps on genome, it explains about that this was generated using information on polymorphism. Polymorphism means, see, polymorphic, that means so many different types of genes are present, and so many different genes, they are going to code for different types of proteins. For this reason it is known as polymorphism, right? So this was generated using information on polymorphism. Now you know that it is occurring in the polymorphic DNA, right? That is polymorphism which is taking place. But whose polymorphism is occurring? That is of the restriction endonuclease recognition sites. See, restriction endonuclease, but before going to the name endonuclease, you need to understand that what is nuclease, endonuclease, right? So restriction endonucleases are the enzymes which are responsible for cutting the vectors, right, as well as the DNA. By these are the enzymes which cut the DNA as well as the vector, right? These two of them should be cutted or should be breaked through one single restriction enzyme. Why is it so? Because they are cutting the DNA as well as the particular vector into same type. Why? Because they're upon the cutting of the DNA or the vector, two types of ends are produced. You are going to study this in detail in while you will study the biotechnology chapters. But here, when you are cutting the DNA, the both vectors as well as the DNA, they both should be cutted into the single manner. That means the same manner they should be cutted, so that they are forming the same types of ends and they can be easily attachable, right? So recognition, restriction endonucleases means on the vector there will be presence of several restriction sites, and these sites are known as these sites are responsible for the attachment of the restriction endonuclease or exonuclease. If it is attaching the with the whole base pair and cutting the whole base pair, that is endonuclease. And if it is cutting only the outer strand or the outer the one strand only, then it is known as exonuclease, right? So you will study this all things in detail, but here you need to understand that what are restriction endonucleases. So restriction endonucleases are the nucleases which cuts the DNA and the vectors into the same manner. And some repetitive DNA sequences known as microsatellites. This also you are going to study in detail in chapter number 11 and 12. So from this you can understand there are that through the physical or the genome, the physical maps which are formed through the help of the genetic and physical maps, you can understand the whole genome in a proper manner. So this is all about the Human Genome Project, and the last topic of the Human Genome Project is the salient features of human genome, right? So the human genome contains 3164.7 million nucleotide bases, that is you have already covered in the previous class. So the human genome, it contains how much nucleotides or the bases? That is 3164.7 million. So this many nucleotides are present, right? Okay, so the human genome, what it contains is, it contains 3164.7 million nucleotide bases, and upon the presence of this all nucleotide bases, you can easily understand the human genome, right? And this is covered inside the Human Genome Project, that is simply said as HGP. Next is the average gene constitutes of 3000 bases. Right now the particular, there is so presence of 20,000 to 25,000 of genes in a human, but amongst them this one gene is containing what? 3000 bases, right? So the whole genome is containing 3164.7 million bases, but amongst them there will be presence of 3000 genes in one, 3000 bases in one gene, right? Next is the third part is the largest known human gene is dystrophin, right, which is containing 2.4 million base. This can be asked in MCQ, that which is the largest human gene. So dystrophin is the largest gene and it is containing 2.4 million base. It is one gene only, but it is containing so many bases, right, that is nucleotides. The third is the total number of genes in human genome is estimated at 30,000. So the human gene contains approximately 30,000 genes. Next is the functions are unknown for over 50% of the discovered genes. So the functions, see, there are so many different types of genes also, but we are not knowing the exact functions of those genes, at what type of function they do in our body, right? So approximately 30,000 are present, but out of that 30,000 we are knowing the functions of only half of the genes, that is of the 50%, right? That is 50%, that comes to 15,000 genes only. We are knowing the functions of 50,000 genes only. Other 15,000, that we are not knowing that what is the exact mechanism or functions they are playing in our body. Next is less than 2% of the genome codes for proteins, right? Now out of this all, only less than 2%, minimum 2% you can take, but less than 2% they are coding for the proteins. So you think that there are so many bases and there are so many genes which are present in your body, but out of them, right, out of 30,000 genes, only 2% of genes they are coding for the proteins. Next is the repeated sequences make up a very large portion of the human genome. Now, there are so many sequences which are repeated regularly, right? That means so many repeated sequences are present in the human genome, and this all the repeated sequences, they have a very large portion which is present in the human genome. So if it is asked that which are the regions or which are the portions which are constantly repeated. So that is known as repeated sequences, right? Next is the repeated sequences that are repeated many times, thought to have no direct coding functions, but they may have a role in evolution, chromosome structure, and dynamics. See, now this repeated sequences, they do not code for any function indirectly, right? That means they are not going for the direct function which is present in your body, but they are involved in evolution. That means whatever evolution is taking place. So if the evolution takes place through the mutation, so once the mutation occurs, then and then only the evolution is possible. So here what happens, the particular repeated sequences, they are responsible for doing what, the repetition, right? And the repetition, they are not playing any direct role in the human genome, but they are responsible for the evolution as well as for the chromosome structures. They are also responsible for imparting structure to the chromosome, as well as they are also involved in the dynamics. So these are the three functions of the repetitive sequences. The next is chromosome I has most genes, that is 298, 2968, and the Y and the Y gene has the fewest. See, this is an example that there is presence of a gene Y as well as a gene I. The gene I in human beings, it is having, this particular gene is having 2968 genes, right? So the chromosome I, which is present in your body, right? It is not located that in which cell is present, but in most of the cells there is presence of different types of chromosomes. But there is a chromosome which is known as chromosome I, and this I chromosome, it has the ability, and it is having number of which a number of genes, that is 2968 genes. While we are talking about the Y chromosome, right, the Y chromosome has the least or the fewest genes, that is 231 genes. So there can be possibility that few chromosomes can have more number of genes and few chromosomes can have few number of genes, right? So this is the difference, that, and it is not it is not common in all the organisms, right? But yes, there is possibility that few chromosomes, they are running or they are playing the same role in all the organisms, right? Now the scientists have identified about 1.4 million locations where single base DNA differences, that is SNPs, that is single nucleotide polymorphism, occur in humans. Now, there are also, the scientists have also identified that about 1.4 million locations are there, and this all the locations, they are single based. That means only single strand is present, and for this reason they are known as single nucleotide polymorphism. So it is not necessary that all the bases or all the sequences will be in double manner. There is possibility that the DNA strands or the DNA or the nucleotides, they can be present in single manner, and it is known as SNPs, that is single nucleotide polymorphism. So this is all about the DNA human genome project. In the next class we will see about the DNA fingerprinting. So now you can turn on your mics and you can ask whether you have any doubts or not.